So, good morning, everyone. So, I'll be presenting my paper on a comprehensive approach to and exploring therapeutic potential in retinal dystrophies. No financial interest. As we know, field of genetics is evolving at a very high pace. Eye is the second only to the brain as an individual organ in its frequency of involvement with genetic disorders, and almost 50% of pediatric blindness is due to genetic etiology. Clinical picture can be sometimes overlapping. It may range from a mild clinical manifestation in case of stationary night blindness to a severe onset early retinal degeneration in case of Leber's congenital amaurosis. So these are some of the complaints with which patient present: night vision problem, normal vision, but tunnel vision. Some have central scotoma, some are allergic to light, they are photophobic. And some parents bring their kids stating that they are not doing well in the school. And some are worried, I have this condition, it's okay, but will my children get affected? So these are the problems what they face. What we are doing as of now is your counseling, genetic counseling we are offering. We are offering low vision services, photochromatic glasses for photophobia, and we are monitoring the disease. So due to its rarity, the clinical models of dystrophy has not been studied extensively in India and needs to be comprehensively analyzed along with the disease phenotypes. Ours was an observational study approved by Institutional Review Board. Informed consent was taken from patient and by guardian in pediatric patients. A total of 45 patients in the age group of 5 to 31 years were studied. There were 31 males and 14 females. All patients underwent comprehensive evaluation, including imaging. For pediatric patient, we used handheld OCT. Electrophysiological tests were carried out wherever required with strict adherence to International Society of Clinical Electrophysiology of Vision. So coming to the genetic testing part, we use next generation targeted sequencing using a panel of 212 retina specific genes, and we look for variant of interest. So we found 45 different pathogenic mutation in all these patients, and some of them were unreported, which are highlighted in the red. So among these, as I told, 9 mutations were unreported. These nucleotide changes were not present in 100 normal controls analyzed. 36 variants were previously associated with retinal dystrophies. So we had 9 novel mutations with existing spectrum of gene mutation, which are from the Indian cohort. So it provides further information on genotype-phenotype correlation for retinal dystrophies. So what does all this mean to patients? So let me explain the same by these two case example. Both these patients presented with blurred vision. Fundus appeared normal clinically. The first patient had a very subtle change at the fovea, but however, OCT picked up subfoveal cavitation. When he did a fulfilled ERG, the dark adapted responses were completely normal, but if you look at the light adapted response, it was unrecordable. The second patient had obvious atrophic changes in the autofluorescence image. Fovea also showed thinning and outer retinal loss. However, fulfilled ERG was near normal, except for maybe a mild reduction in dark adapted responses. Why there is so much of functional difference, though both look similar clinically? The reason being the first case is a case of achromatopsia caused by GUCY2D mutation gene. And this is a stationary disorder. Patient is born like that and is going to live like that. And we need to counsel and we need to advise the career according to his needs. The second case is a Stargardt's disease. This patient had a mutation ABCA4. Though it may look functionally good now, it is likely to progress by taking some measures like avoiding bright light, avoiding vitamin A supplements. To extent we can arrest the progression. So coming to the future, we know future is definitely going to be dominated by gene therapy for dystrophies. Basically, gene therapy involves an injecting adenoviral vector, which carries a corrective gene, which gets inserted into the host cell where it's expected to function normally. So how to produce this AAV? It's a very complex process where plasmids will be generated first, which will be transfected into HKE cell lines. These will be harvested 65 hours later, and this will undergo multiple purification process, finally resulting in an AAV particle. This is the electron microscopy view of the real AAV particle. So we built a mouse retina model where we injected subretinally into this mouse model of the lag gene, which codes for beta galactosidase enzyme, and a separate ethical clearance was taken for this animal study also. This is the fundus photo of the mouse. We can see the injection site, and we also used fluorescent bead tracking system just to see whether they have properly migrating in the subretinal space. So 2 months post-injection, these eyes were harvested. And if you look at the negative control eye and the injected eyes, there was a very good staining of the beta galactosidase, which is quite promising. That means the uptake is very good, whatever the vectors we injected. So I would like to conclude by under these headings. What we knew so far was diagnosis of dystrophy means almost end of the road to patients. What this study adds is more database, and we had 9 novel mutation. It's indicating that somewhere the Indian database for mutation may be different from rest of the world. We need our own database. And subtle changes, as I showed in those two case example, can make a big difference in the patient management and how we counsel. Rodent model was also studied in our study. The future will obviously be to carry out the first in India human gene therapy. Thank you so much.
Thank you, Doctor. If there are any questions, we'd like to take them now.
Dr. Purnachandra, you mentioned in your last slide that you would like to, create a greater database the Indian genome and the Indian defective genes, so far as all these retinal dystrophies are concerned. So towards that, what is your institution doing?
So as of now, like all the patients which have come to the dystrophy clinic, first they will be obviously undergo thorough clinical evaluation just to confirm that it is dystrophy, not something else which is treatable conditions. And these patients undergo a thorough genetic counseling first, just to make them understand what exactly genetic testing is going to offer. After that, there is a cost limiting factor when it comes to genetic testing. So as of now, as a study part, we are doing all this for free from our institute for all these patients, and we have generated data for almost more than 1,500 dystrophy patients. Eventually, since I mentioned the next generation targeted sequencing, once we have this large data, we can do a specific mutation screening, which will bring down the cost like anything. For that, we need to have this data, what we have generated.
No, but are you also parallelly collecting and screening normal people and building up the normal database?
Yes, there is an exact genome database which is called, in which we have collected all the normal database. Once we generate any report, we have to look for the significance. For example, if we detect one mutation, we need to look at the frequency of that mutation happening in a normal population. So then only we'll compare with our mutation becomes significant.
But that
Otherwise,
means
if I undergo genetic testing, I may have some mutation. The issue is how significant it is. We have taken care of that part.
But that means you must have collected, against 1,500 people with retinal dystrophies, you probably must have collected what,
Yes,
15 lakhs of normals? Or
no, not 50 lakhs as of now, sir. Of course, in India we do have some logistics. It's around a lakh. It's around a lakh. Okay. But and also one more thing, sir. This is our thing what we added. But there's in the past also exact genome database they've already collected. It's not from our institute. In the genome, whole exome genome, there is a database for Indian patients also.
Yes.
That's our data.
So you probably need to tie up with those centers where they already have
Yes.
the
genome. We do have tie-up with Moorfields Hospital, and all of them have approached, because once we presented our Indian data, they were very much happy to see this variety of mutation, novel things and all. Even they were very happy to collaborate, sir.
Manoj, please ask your questions.
Yeah, Pooran Chandra, good work as usual. Just two comments or maybe suggestions. Can we extrapolate this data to most of the retinal dystrophies? That is one. And again, comparison of this data from Indian subset as well as overseas data should be, is the need of the hour. Then only we'll have more light on this particular research as how we are going to take this forward.
Yes, I'll answer that. So the first part you told, definitely this data can be extrapolated, because if you know that whenever we report the genetic, they'll be reported as pathogenic or variants of unknown significance.
Absolutely agree.
Yeah, once it is reported pathogenic, automatically it becomes relevant to other dystrophies also. And the second part, when you told, which is a very good point which you made, that if you look at if you comparable with the Western data. So here we need to compare it, because for Indian database for mutation, I told it may be different. For example, if someone designs a gene therapy model for something in the US or Moorfields, we can't come directly and inject that patient. Here the mutation may be slightly different. For
that
example,
is where the challenge is going to be.
Exactly. It's a good point you made. Thank you.
Purnachandra, before we conclude, just one comment that please do a good spell check of your presentation. The spelling of achromatopsia was wrong. So okay, similarly, check out all your spell checks. Okay, go through all the slides again. Okay, sir, okay. We won't hold it against you for that spelling mistake.
I didn't. Okay, thank you, sir.